marvelous. How perfect. Oh, the devil take it. They're mongrels. No spots. No spots at all.
Yep, that's me. You're probably wondering how...
Never mind. Today we're making Dalmatians that glow. Let's do it. Okay, so you might be wondering how we aim to induce the glow-in-the-dark characteristic into dogs, and chances are that you already know the answer. If you've ever been to a zoo with a large aquarium, or if you've ever seen a nature documentary that for once doesn't involve Sir David Attenborough telling you how animals have sex, you would know that corals, sea anemones, some bacteria, and jellyfish contain this characteristic. This is because these animals contain a protein known as GFP, or green fluorescent protein, which under lights of lower wavelengths than green light appears to fluoresce green, as implied by the name. Commonly, GFP is used in experiments involving the transformation of bacteria like E. coli, where new traits are introduced to them using plasmids. In these experiments, to see whether the plasmid has been taken up by the competent bacterial cell, the gene for GFP is inserted into the plasmid along with the gene of the trait being transferred. If you're more curious about this, there's a link in the description that you can read. But put simply, once bacterial cells have taken up the plasmid, they should fluoresce green under blue light, or lights that are of lower wavelengths than green light, which suggests that they also contain the other trait of which the gene was present in the plasmid. This may include antibiotic resistance like that to ampicillin. Simple enough, right? Well, we're actually not the first people attempting to do this. Here's the GFP bunny. And as a side note, the controversy surrounding this is absolutely insane. The artist that commissioned a lab to make the bunny wanted to take it home, and also when he was informed that it had died, he thought the lab was lying to him, as if a lab would murder or even lie about a bunny having died just to prevent a sick art piece. Anyways, unlike the bunny, which according to Wired looks like a bunny that was dipped into a bucket of paint, we're attempting to make the Dalmatian fluorescent only where it has black spots in its coat. For this, we need to first understand why Dalmatians even have the coat that they do. Put very simply, it's because they have this gene known as the piebald gene, and this causes a mutation in another gene known as the MITF gene, the full form of which is melanocyte-inducing transcription factor. That's a mouthful. But essentially, this gene codes for a protein that is a transcription factor involved in the development of melanocytes. That sounds tough, but put simply, melanocytes are cells that produce melanin and thus pigment. So MITF being mutated essentially disrupts the development of these cells that produce melanin, and as a result, these cells can't migrate to the skin in Dalmatians, and that's why you have a white coat with black spots on it. And all you need to understand, for the sake of this experiment at least, is that the black spots are where melanin and melanocytes are present, whilst the white is where there are no melanocytes and thus no melanin. So, coming back to our goal of having Dalmatian spots be fluorescent, what we need to do is have GFP be expressed where the Dalmatian has these melanocytes inside the melanocytes. So for that, we need to understand how promoters and gene expression works. And you might have wondered this independent of this video, that why is it that despite every single cell having the same DNA, they still have distinct functions? Well, that's because of the switching on and off of genes as a result of promoters. To better understand that concept, it helps to think of promoters as doors into various clubs, and transcription factors as their bouncers. So essentially, once the bouncer allows RNA polymerase to enter the club through the door by binding itself to the promoter, that results in the transcription of the gene attached to the promoter, and thus later the translation. But I guess the equivalent of this would be to go into a club and take pictures of people, which do not do that. So if we were to take this analogy further, every single cell in the body has these various doors to different clubs, yet only specific cells, with regards to their function, has the specific bouncers for these various clubs. An example is of the beta cells in the pancreas, which produce insulin and thus express the insulin gene. These cells not only contain the promoter and the gene for insulin, as does every other cell in the body, but they also contain the specific transcription factors that enable the expression of the insulin gene and thus the production of insulin. Coming back to our goal, in order to produce green fluorescent protein only in the melanocytes, we need to first identify a gene that is expressed only in melanocytes, choose its promoter, attach its promoter to the GFP gene, and then deliver that DNA to a Dalmatian embryo. An example of a gene that is expressed only in melanocytes is the TYR gene, which produces the enzyme tyrosinase, which catalyzes the first step in melanin synthesis. If we were able to isolate the promoter for the TYR gene and attach to the promoter the gene for GFP and insert both of these using a virus into a Dalmatian embryo, hypothetically, we would be able to induce the production of GFP only in melanocytes, because when the transcription factors are produced and bind to the promoter for the TYR gene to produce tyrosinase, they will also bind to the promoter that we have inserted in the embryo of the Dalmatian and thus bring about the production of GFP as well. Admittedly, our use of GFP wasn't the most helpful to the greater population of the world, but that does not mean that there are not examples of GFP usage in the real world that are benefiting both the health community and also the advancement of this field of biotechnology. These include many attempts to visualize the spread of cancer in animals using GFP as a marker and many other such uses. If you enjoyed this video and learned anything at all, I'd really appreciate if you could like and subscribe, and I'm hoping to produce more videos like this soon. Thank you for watching.